Hello everyone, welcome to Venom Guidance. This is Dr. Anuja Sharma, and in this video we'll discuss transposable genetic elements or transposons. So let's start the video. But before we get into this video, I have some very important piece of information for you. As you know, the NET exam is almost here, and you all need some serious practice before you sit down for the real exam. Well, guess what? Uncademy is here at your rescue. Yes, Uncademy is organizing a free Revise India series for CSIR UGC NET November 2020 exam. And I repeat, it's free. This series will include more than 350 free classes from India's top educators, including CSIR UGC NET faculties, toppers and mentors. Now, this is a great opportunity for all the students to prepare and analyze their progress through Uncademy free revise series. So I highly recommend that you attend this series, as this will give you that last minute revision before the exam and will definitely boost your confidence. Now, because the more you practice, the more you learn, and the more will be your chances to crack CSIR exam. And well, it's free, so that's a bonus. Now the series will begin on 1st November and continue till 15th of November. And during the span of 15 days, there will be more than 120 hours of free live classes on Uncademy. So you can choose your subject and learn about the best techniques to crack the exam. So please don't wait. Click on the link in the description box and choose your special free class today. All the best to all the students for their exam. Let's crack it. Again, the link is in the description box. Click it, share it. So let's start the video now and talk about transposons. Transposons or transposable genetic elements are DNA sequences that can move from one position to another. Now, these transposable elements or transposons are also known as jumping genes. And the process of movement of transposons is called transposition. Now, when transposable elements move from one location to another, they may break chromosomes or mutate genes. Now, also when they move from one position to another, it can be in the same chromosome or another plasmid. There are basically three types of transposons, and most of the transposons can be classified into one of these categories based on how they transpose. The first one is conservative or non-replicative. Second is replicative transposons, and the third one is retrotransposons. Now let's talk about the first category of transposons, which is the conservative transposons. Now, in this first category, transposition is accomplished by excising an element from its position in a chromosome and then inserting this element into another position in the same chromosome or in another chromosome or another plasmid. And this excision and insertion events are catalyzed by an enzyme called the transposase, and this enzyme is usually encoded by the element itself. Now the transposons are liberated from the donor site by a double strand break in the DNA. So this mechanism is also referred to as cut and paste transposition, because the element is physically cut out of one side in a chromosome and pasted into a new side. Some of the examples of conservative or non-replicative transposons are IS elements, which are present in bacteria, composite transposons, for example Tn3, which are also present in bacteria, ACDS elements present in maize, and P elements of Drosophila. Now next is the replicative transposons. In replicative transposons, transposition is accomplished through a process which involves replication of the transposable element's DNA. So the transposase, encoded by the element itself, mediates an interaction between the element and a potential insertion site. And during this interaction, the element is replicated. So one copy of the element is inserted at the new site and the original copy remains at the original donor site. Replicative transposition involves only single strand breaks of the DNA at the donor site, unlike the conservative or non-replicative transposons in which double strand breaks occurs at the DNA of the donor site. An example of replicative transposons is Tn3 element, which is present in bacteria. Third category of transposons is retrotransposons. In retrotransposons, the transposition is accomplished through a process that involves the insertion of copies of an element that was synthesized from the element's RNA. Now, an enzyme called reverse transcriptase uses the element's RNA as a template to synthesize complementary DNA strand, and from this complementary DNA strand, a double-stranded DNA is synthesized. Now, this DNA is then inserted into the new chromosomal sites. Because this mechanism reverses the usual direction in which the genetic information flows, that is, the genetic information flows from DNA to RNA. However, in this mechanism, it is flowing from RNA to DNA. So this kind of mechanism is referred to as retrotransposition. And the elements in this category are called retrotransposons. Now, some of the elements in this category are related to a special group of viruses, that is the retroviruses, and thus they are called retrovirus-like elements. Other elements that engage in retrotransposition are simply called retroposons. So these are the three main categories of transposable genetic elements or transposons. Now next we'll discuss the transposable elements in bacteria. But before we move forward in this video, I request you to please subscribe to this channel and hit the bell icon for future updates. So let's talk about the transposable elements in bacteria. And the first one that we'll discuss is IS element. The IS element was the first transposable elements discovered in bacteria. And it is a conservative transposon, meaning the transposition process is accomplished by excising an element from its position in a chromosome and then inserting it into another position. These are the simplest bacterial transposons. Now, IS elements were first detected in certain lac mutations of E. coli. A typical IS element consists of fewer than 2500 nucleotide pairs and contain only genes whose products are involved in assisting or regulating transposition. So the IS element consists of a central region of about 700 to 1500 base pairs, and this central region is surrounded by a short identical or nearly identical sequences of about 9 to 40 base pairs at its end. And this number that ranges between 9 to 40 base pairs, it depends on the specific IS element. These terminal sequences are called terminal inverted repeats, as they are always in inverted orientation with respect to each other. The central region of the IS elements contain DNA sequence that act as the transposable element and also has genes that encode enzymes involved in transposition, the transposase enzyme. And inverted repeats, they carry the transposase recognition sequences. Now to these sequences, the transposase enzyme binds and then cuts both strands of the DNA. This cleavage excises the element from the chromosome or plasmid so that it can be inserted at a new position in the same or separate DNA molecule. IS elements are therefore known as cut and paste transposons. Now when IS elements insert into chromosomes or plasmids, they create a duplication of part of the DNA sequence at the site of the insertion. And one copy of the duplication is located on each side of the element. These short directly repeated sequences, which are around 2 to 13 nucleotide pair long, are called target site duplications. And these target site duplications arise from staggered cleavage of the double-stranded DNA molecule. Now let's see in detail how target site duplication actually occurs. Now if you look at this picture, you'll see that transposase enzyme makes staggered cuts on the target DNA. And after these cuts are made, the transposon is transferred and inserted at the target site. Now this leaves gap on both the sides, which is later on filled with polymerase enzyme. And this results in the formation of direct repeat on both the side of the transposon, resulting in target site duplication. Now next we'll talk about composite transposons. Composite transposons are created when two IS elements insert near each other. So the DNA sequence between the two IS elements, which was otherwise immobile, can now be transposed when the two IS elements act together. A composite transposon consists of a central region surrounded by two IS elements. The region between the IS elements in each of these transposons contains genes that have nothing to do with transposition. The central region usually contains genes that confer antibiotic resistance. For example, Tn3 contains the genes for transposase and resolvase enzymes and also has tetracycline resistance gene. IS elements at the two ends of a composite transposon. They can be identical or different. Like in Tn5, the element on the right, called IS50R, is capable of producing a transposase to stimulate transposition. But the element on the left, called IS50L, is not. This difference is due to a change in a single nucleotide pair that prevents IS50L from encoding the active transposase enzyme and an inhibitor protein, but instead results in the synthesis of P3 and P4, which are non-functional analogues of transposase and inhibitor. Now, another factor to consider is that the IS elements can be in the same or different orientation. Like in Tn9, the flanking IS elements are in the same orientation with respect to each other, whereas in Tn5 and Tn10 elements, the orientation is inverted. And these different orientations are designated as L for left and R for right orientation. Also, composite transposons, like the IS elements, also create target site duplications when they insert into a target DNA. Next, we'll talk about Tn3 transposons. Now, Tn3 transposon is a replicative transposon and it does not contain any IS element, unlike Tn5, Tn9 and Tn10. Tn3 transposon is 4.9 kilobase pair in size and terminate in simple inverted repeats, which are 38 nucleotide pairs long. It contains three genes, TRPA, TRPR and BLA. TNPA encodes a transposase enzyme, TNPR encodes a resolvase or repressor enzyme, and BLA encodes an enzyme called beta-lactamase. This beta-lactamase confers resistance to the antibiotic ampicillin, and the other two proteins, which are transposase and resolvase, they play important roles in transposition. Now, transposition of Tn3 transposon, it occurs in two stages. During the first stage, the transposase enzyme mediates the fusion of two circular molecules, for instance, two plasmids, one carrying Tn3, the donor plasmid, and the other not carrying it, which is the recipient plasmid. And this resulting structure is called a cointegrate. Now, during the formation of cointegrate, Tn3 is replicated. Now, one copy of the Tn3 is inserted at each junction between the original replicons, that is, where the two plasmids have fused. And these two copies of Tn3 are oriented as direct repeats. During the second stage of transposition, which is also known as resolution, the TNPR encoded resolvase or repressor mediates a site-specific homologous recombination event, which occurs between the two copies of the Tn3 transposons present on the cointegrate. Now, this event occurs at a sequence in Tn3, which is called the resolution site. This event releases two constituent plasmids, each of which has a copy of transposon. One is the original donor replicon or plasmid, and the other is the target replicon or plasmid that has gained a transposon. And this transposon in the target replicon or plasmid is flanked by a short direct repeats of the host target sequence, also known as site duplication. That's all in this video. If you have any queries or questions, feel free to leave them in the comments. Thank you.